start with Danielle Belgrave. Danielle is a researcher at Microsoft Research Cambridge in the Healthcare Machine Learning Group. She also holds a tenured research fellow at the Imperial College of London. Her research focuses on developing probabilistic and causal graphical modeling frameworks to understand disease progression over time. The aim of the research is to use machine learning to identify distinct subtypes of disease evolution and to understand the underlying mechanisms of these subtypes so as to develop personalized disease management strategies. She has a BSc in Business, Mathematics and Statistics from the London School of Economics and a Master in Statistics from University College London. She was awarded as a Microsoft PhD scholarship to complete her PhD in Statistics and Machine Learning Applied to Health at the University of Manchester. She received a Medical Research Council Career Development Award in Biostatistics for the project Unified Probabilistic Latent Variable Modeling Strategies to Accelerate Endotype Discovering in Longitudinal Studies. Please, we have a warm welcome to Danielle.
Thank you so much, Feifo, for that kind introduction. Let me just see if I get this up. Okay, good morning, and thank you so much for this invitation from the organizers to give this presentation about a topic that I'm really passionate about, and I hope that it's contagious. And so I'm going to talk about machine learning for healthcare and some of the opportunities and challenges around machine learning for healthcare. So why is healthcare such an important topic at the moment? So in the United States, over the past 8 years, the volume of electronic healthcare records has increased by 9 times. So there's a huge boom in the amount of electronic, readily accessible data available. And there's been a move in the United States as well towards publicly available open access data sets. So this is one of the biggest data sets in the US, which was born out of MIT, called MIMIC. And any researcher can have access to MIMIC. And it's the electronic healthcare records of millions of patients across the US. And there's a big push in MIMIC towards collaborative and open science. So MIMIC is an openly available data set developed by MIT labs. And it comprises of 60,000 intensive care unit admissions. And it includes data on demographics, vital signs, lab tests, medications, and much more. So we have access to these rich data sets. Another reason why machine learning for healthcare is such a huge opportunity at the moment as well is that, well, we've had a lot of talks on CNNs and the use of GANs, and most of the successes in machine learning for healthcare have been in image recognition. So this is an example of using convolutional neural nets which can scale bigger data sets. So this is an image of for cancer to inform radiology treatments. And using CNNs, we can have state-of-the-art image segmentation to detect where radiologists should target treatments. So I'm not going to talk about convolutional neural nets and images. I'm going to focus more on the messiness of healthcare data and some of the challenges around that. And I'll motivate these in examples. So one of the huge opportunities of machine learning for healthcare is the diversity of digital data that we've seen in the past 10 years. So there's a lot of data available and available electronically, but also available automatically. So there's a lot of data in the wild. People wear Fitbits, smartphones, smartwatches, and there's a proliferation of the types of blood tests that are available. We can think that within the past 5 years, the cost of genetic screening has decreased. I think you can get your genetic profile for $100 or $50 maybe, and you can get predictions of what diseases you're likely to get. And there's a huge proliferation of lots of different types of data, image data, electronic healthcare data, GP records. But with this comes many challenges, and there are lots of challenges in machine learning for healthcare that we haven't solved. So I'm going to focus on 3 of these challenges, but these are what I see as the main challenges in machine learning for healthcare. So the first challenge in machine learning for healthcare is that we have heterogeneous patient populations. So very often when a drug is developed for patients, it's developed with assuming particular characteristics of patients. So you assume a homogeneous population, but what we see in reality is that populations are actually heterogeneous. The other related to this problem is the problem of generalization. So if you develop drugs or if you have study findings for one particular population, how do you generalize this to another population? Causal discovery is a big problem and it's a big challenge as well. So I'll touch on this in the talk as well. Data in the wild, they are heterogeneous data types, and one of the huge challenges in machine learning for healthcare is how do we merge different types of data? How do we merge genetic data with clinical data in order to get causal discovery? The reality of healthcare data, which doesn't exist so much in other types of data, I think, is missing data. And very often, healthcare data relies on people answering surveys, answering questions. So not just missing data, but also measurement error as well is a big problem in machine learning for healthcare, because if you're asking patients for information, how do you know that they have a good assessment of how they feel or of the reality of their symptoms as well? And 3 other big challenges, which I'm going to allude to, but I won't go into detail on, interpretability. So once you have a good model, how do you make those findings interpretable and transparent? Fairness as well is a huge challenge in machine learning for healthcare. Once you develop something, you want to be in a situation where you can deploy it in the wild. And how do you make sure that we're not introducing any biases? And I think a huge challenge is a trade-off between accuracy, so getting a really good model, and fairness. How do you make sure that by getting a really good model, you're not marginalizing certain groups? So I'm going to motivate most of these challenges within a few examples, some of the examples from my work, and some of the examples from the work of others as well. So the first challenge, which I think is the biggest challenge in machine learning for healthcare, is heterogeneity in patient populations. So as I mentioned before, typically when you develop a drug or you develop some intervention for machine learning for health, using machine learning for healthcare, the assumption you make is that you have a patient group. So this could be patients with lung cancer, let's say, and these people have the same diagnosis and the same prescription. So typically, in a pharmaceutical trial, you develop a drug, and you want to test that drug on the patient population. So in order to estimate treatment effects, which I think is one of the huge, one of the basic challenges in not just machine learning for healthcare, but statistics for healthcare, you have your patient group, and you split the population into 2, and you randomly allocate the 2 groups some drug. It could be drug A or drug B, and you want to compare how many people are cured using drug A and how many people are cured using drug B. So you use some test statistic via using supervised learning models or regression, and we measure the outcomes for both. So we get the proportion of people who are cured from drug A and the proportion of people who are cured from drug B, and we do a comparison of the 2 groups. But the post-marketing reality is very different. So what we see is that same patient group of people with lung cancer, we see that drugs affect people very differently. So for some people, the drug is beneficial and it has no side effects, and these are the people who we want to give the intervention. But there may be some people who have negative side effects even though the drug is beneficial. So in that case, what we'd want to do in an ideal world is to measure the benefits-risk ratio. So that's a huge study in pharmaceuticals, trying to figure out, is it really worth giving someone an intervention because of the potential harm? And this is very prominent precisely in cancer trials, where in cancer post-marketing trials, where maybe sometimes patients get really bad side effects and it's not worth giving them the drug because it doesn't prolong, it's not effective, and people have really bad quality of life. For some people, the drug just doesn't work, and we want to see who are the people that the drug doesn't work for. So this has led in machine learning to healthcare to a huge study of endotype discovery. So endotype discovery is a big challenge in machine learning for healthcare. And what we try to do in endotype discovery is to discover who are the people who would respond to treatment and who are the people who won't respond to treatment. And this is very much looking at symptomology to see whether we could identify that heterogeneity in disease through looking at how patients manifest symptoms over time. So this is what I'll talk about in the first part, as a way of motivating how can we understand patient heterogeneity using probabilistic graphical models. So typically we want to identify subgroups of complex diseases because not everybody shows symptoms in the same way at the same time. And the purpose of trying to do this is to see whether we could identify underlying causal mechanisms which are distinctive for these different subgroups. So could we identify early on people who would respond to treatment and people who won't respond to treatment? And this is the foundation of stratified medicine and for seeking better targeted interventions. So 2 examples that I've been working on just to motivate this at a high level at first is heterogeneity and asthma. So typically asthma is an umbrella term to describe many symptoms. So we can imagine children going to a clinic at the age of 5, and they have these different symptoms. So you have allergy, poor lung function, you take asthma medications, and they're all told you have asthma. But what we see outside of the clinic over time is that some people may grow out of asthma, some respond to treatment, some don't respond to treatment, some have different levels of severity, or some may develop asthma later on in life. So the question is, are these really the same disease? Is this really all asthma? Or are they different endotypes with different causes? Another area I've been interested in recently is the area of mental health, where you have heterogeneity of symptoms. And we may see people with these different symptoms, and they may be given a diagnosis of depression. But depression is a very heterogeneous condition, so looking at the manifestation of symptoms over time, can we identify different subgroups of mental health and see whether there are different interventions for different types of mental health conditions? So the general framework for looking at endotype discovery is through using a probabilistic graphical modeling framework to identify different subtypes of disease. So the motivation is you have a patient who has different phenotypes or different observable symptoms. So this would be your F1 to F8. And this could be, for example, in the case of asthma, you come in with allergy, you have really poor lung function, and you're wheezing. So you have these different features over time. They could also be features of genetics and different things. And what we want is a parsimonious description of the data to see how people develop symptoms over time and whether we can group people into different subgroups inferred from what's observed. So these would be our X1s, X2s, and X3s. And these are latent variables which aren't directly observed, but which we could infer from these phenotypes or our observations. So the general tool we use for this is a probabilistic programming tool for identifying latent structure in the data. So in this, we have some probabilistic model, and the probabilistic model is generally what we assume about reality. What's the current knowledge of a situation? And this could be developed through talking to collaborators, clinicians, or through reading papers as well. So we have some baseline knowledge, and we use that as our probabilistic model. Then we have some inference algorithm, which we want to answer different queries. So this could be, are there different subgroups of patients? How many subgroups of patients are there? And we have evidence, which is the data, and we use the evidence to infer these different queries or questions that we have. And then we get a probabilistic answer, and that gives us probabilities of different outcomes. So it could be probability that a person belongs to a particular subgroup, or probability of someone going on to develop asthma in that case. And the general ecosystem for doing this is probabilistic programming. There are several different ecosystems, like Inf.net, which was developed in Microsoft, Edward, Pyro, which was developed in Uber Labs, and Stan. These are all ways of doing probabilistic programming. And you put in your observed values, your prior probability distributions, and you get out some posterior probability distributions. So I'm going to motivate this with a few examples from the literature. So the first example is going back to the example of asthma, where we observe different symptoms for different subgroups of people, for different people in the clinic, and we all give them a diagnosis of asthma. So using probabilistic modeling, what we want to see is, can we identify these different endotypes or these different subgroups of patients? So using data from the Manchester Asthma and Allergy study, which is a birth cohort of 2,000 children, so these children were recruited at birth, and they were followed until age 8. And the purpose of this study was to see, how do people develop asthma over time, and is it a heterogeneous condition? So using data, we used data from their GP records, so people went to the doctor, and the observation was, has this person wheezed or not? Also from the GP records, looking at things like medication, so has a child received medication, what medication have they received? And also we use parental observations as well. So at each year, asking parents, has your child wheezed or not? And we use the longitudinal logistic regression model, where we assume that the probability of observing one of these symptoms at a particular time point is weighted by where have you gotten this piece of information from, so who rates the person as having a symptom, gender, because there's difference between boys and girls in lung development, and the third one is age. So we assume that there's some latent class, which is your true current wheeze, and we want to infer how many subtypes of wheeze are there in different subgroups. So doing this, we identified 5 different patterns of wheeze over time. So this was the optimal solution when we looked at the model evidence. And just at a high level, what you see are 5 distinct subtypes of asthma over time. So it's a population birth cohort, so most people don't have asthma. That's 50% of the population. But what we see is different profiles over time of how people develop asthma over time. So you have people who start off wheezing and then grow out of asthma by age 5. And you have these 2 groups that are persistent asthmatics, but we have a small group of 3%. So this is 13% of the population and 3% of the population who have different characteristics. And it's really interesting that these 2 groups, even though they're classified as classical asthmatics, have different triggers, different allergic responses, and also different genetic profiles as well, which I won't go through. But the interesting thing as well is that by doing this, we can distinguish response to treatment. So that same group of people who wheeze all the time and have severe asthma symptoms, who we call persistent troublesome wheezers, this shows their, the amount of treatment they have, so they are taking treatment over time. But this is the amount of hospitalization for asthma. So these are severe asthma events. So this group, even though they're receiving medication, they have controlled asthma, whereas this group, they don't have controlled asthma. So using these strategies, we could distinguish different subtypes of asthma and also different genetic characteristics and possible underlying mechanisms as well. And also, we could identify who would respond to treatment and who won't respond to treatment. So this is a particular study that's actually had some impact in the pharmaceutical industry as well, in discovering different underlying characteristics for these patients who don't respond to treatment. Another motivation for using this endotype discovery via probabilistic models is getting early interventions for public health, from a public health perspective. So here we have lung function over time, and what we notice is that we could identify these persistent troublesome wheezers. So these are, the higher your lung function, the better your lung capacity. And these are people who tend to be asthmatics. And also, there are people who tend to be more exposed to smoking, or people with good lung function tend to be less exposed to smoking. So in conclusion, what we discovered in this particular study is that asthma is a heterogeneous condition. We have people who wheeze early and then grow out of wheeze, people who develop asthma later on in life, and people who are persistent asthmatics. But also there are different types of persistent asthmatics. And these groups have different genetic and environmental associations and also different response to treatment. Another way of disentangling heterogeneity or looking at heterogeneous populations is through trying to understand particular possible causal mechanisms of how people develop from one symptom to another. So I'm just going to motivate this in another study that we've done, and this is the last study I'm going to show of the work that I did while I was at Imperial College. So with this, we used two different populations, one from Manchester, the previous study I showed, with 2,000 children, and another from Bristol with 10,000 children, so two geographically different conditions. And the hypothesis we're trying to test here is something called the allergic march. So the allergic march is a hypothesis used to explain how symptoms of allergy develop over time. So the hypothesis is that in early life you have eczema. So if someone has eczema, they're more likely to develop asthma, and then they're likely to develop hay fever or rhinitis later on in life, so allergies. And this is really important because these symptoms were seen to be causally linked for a very long time. And because they're causally linked, a lot of work has been done in targeting interventions early for eczema. So the hypothesis is that if you target children who have eczema in early life, you can stop them from developing asthma and hay fever later on in life. So using this data from these two populations, we wanted to see whether we can capture disease heterogeneity and whether this trajectory of symptoms actually exists on an individual level, or whether there are different patterns of symptoms over time. So we looked at probabilistic dependencies to see whether we could identify meaningful latent structure and dependencies. So we assumed some Dirichlet prior distribution with equal probability assignments to different numbers of latent classes. And we assume that each person has an equal probability of belonging to a class, and also that there's equal assignments to latent state membership. So for each of the children in these two cohorts, we calculate their posterior probability of belonging to a particular latent class and assign them to their most likely probability. So we use three models here. These are based on hidden Markov models. The first was where we assume that eczema, asthma, and hay fever or rhinitis are independent conditions. So you assume that the probability of having eczema at age 1, so the probability of having eczema at age 3, for example, is conditioned on what happens at the previous time point, and that this transition of symptoms over time is governed by some overarching latent class of eczema. And we assume the same for asthma and hay fever as well. And we assume that these three independent transitions is governed by some overarching latent variable, and that we can assign each of these children to one of those latent classes, and we assume between 2 and 15 latent classes. The second model was looking at an explicit model formulation of the allergic march, where you condition the probability of wheeze at age 3, for example, on what happens at the previous time point. And the same for rhinitis. You condition the probability of observing rhinitis at age 5 is conditioned on the wheeze status at the previous time points. In the third model, we just assumed a longitudinal latent disease profile, where at each time point, your probability of disease is independent. So you have eczema, wheeze, and rhinitis at a particular time point, and you have some latent state of disease. And we assume that this transition is governed by some overarching latent class. So then we compared these three models, the independent conditions model, the allergic march model, and the independent conditions across time model, using model evidence. And also we looked at So what we found is actually this model gave us the best model evidence. And when we compare different priors as well, we found that using an 8-class solution, so we assume there are 8 different states of symptoms over time, gave us the best fit as well. And this was quite robust to using different pseudocounts for the prior distribution. So what we found when we entangle those 8 classes, we found that only a small proportion of children follow something similar to this, which is our allergic march. So even though on a population level, if you look at the population level data at any given time point, you see something like this. If you look at an individual profile over time, you actually see that most children have wheeze by itself, so this is 8%, or eczema by itself, which is 15%, or rhinitis by itself, which is about 10%. And only 3% of children follow something similar to allergic march. So this is really important in understanding differentiating mechanisms for different subgroups of patients. The third example is taken from some work by Suchesaria's lab, looking at heterogeneity in complex chronic diseases. So in this study, the aim is to predict future trajectories of lung function over time. So scleroderma is a complex condition which affects the lungs, but it also affects multiple organs as well. So it affects the heart, the epidermis, and also the kidneys as well. So in this study, they're looking at, the motivation is to identify heterogeneity in scleroderma, which is a complex chronic disease, and to see whether you can predict lung failure to intervene early over time. And this is a challenge because not everybody with scleroderma will have poor lung function, but the people who have whose lungs collapse, you want to intervene early to prevent that. So there's this motivates trying to predict that event early on and seeing whether we could predict it on an individual level. So similarly, what they used here is a probabilistic model where they assume 4 factors of variability. So they assume that there's some general population level variability, which you try to capture. Then there's a subpopulation variability. So this could be disease subtype. So looking at multiple organs, can you identify these different subtypes of disease? And then there's the individual level. So there may be confounders, such as if someone smokes or not, which has an effect on individual level scleroderma. And there's some sort of structured noise as well. So they've broken this up, and I strongly recommend this paper. It's really a clearly written paper on looking at using probabilistic models for identifying subtypes of disease. So they break this down into these 4 groups: the population level variation, the subpopulation level variation, individual level, and some random noise. And this is the structure that they use, the graphical model, and the different boxes indicate different levels of variability components. So I won't go into detail of explaining these, but the Y is the lung function that we're trying to predict, and we have some function of, it's a function of time, and also a function of different features, which are governed by some latent feature. And what they found is that they were able to predict different subgroups of patients. So this is if you use a population level model, so it fits the curve quite badly. If you include some subpopulation latent variable, it fits a bit better. But then using individual characteristics, you get a much better fit and a much better prediction of lung function over time for these 4 patients. The next challenge I'm going to talk about is one of the huge challenges of healthcare data, in particular electronic healthcare data and questionnaire data, which is the challenge of sparsity and missing data. So missing data, I strongly recommend investing time in understanding missing data. Can I ask how many people work in machine learning for healthcare or work in healthcare applications? I saw quite a few posters yesterday, yes. So I'm sure this is something you're all familiar with. And I think this is the first thing we should do when looking at any healthcare data. Look at investigating mechanisms of missingness and really trying to understand what is happening in the data set. Is there missing data? Because this can bias our results. And there's a fantastic piece of work by Alexina Mason and Nikki Best looking at strategies for modeling non-random missing data mechanisms for observational studies using Bayesian methods. And I think, so a lot of the work that I've presented before, and also the missing data work, the advantage of using probabilistic graphical models is you can have a unified framework. And in this work, they show a unified framework of using probabilistic graphical models, also for compensating for missing data as well, in a very heuristic way. So there are three main assumptions we can make about missing data. So these assumptions were developed by Donald Rubin in his classic paper on missing data. So the first assumption is that data is missing completely at random. So under this assumption, the probability of data being missing doesn't depend on anything in our data set. So an example with this would be if you have blood samples and you dropped a blood sample. It's probably just random, the reason you dropped a blood sample. So here, the way we model that is we say the probability P of the data being missing is some constant. It's a half if you've dropped half the samples, for example. The second mechanism of missingness is we can say data is missing at random. So in such a case, we say that the probability of data being missing doesn't depend on anything that's unobserved, but it's conditional on something you've observed in the data set. So an example would be, from the previous study that I showed, would be children with missing wheeze data have better lung function. So if we have the lung function data, we can predict the probability of a child wheezing. So in this example, the probability of a child wheezing or not would be dependent on some constant plus another constant weighted by your value of lung function. Or it could be a function of time as well. So the older someone gets, the less likely they are to participate in a study. So the probability of their data being missing is a function of time. The third case, which is a lot harder to model, is when data is missing not at random. So in this case, the probability of data being missing depends on something you've not observed. So an example of this would be a child with missing lung function data has better lung function, so they're less motivated. But if you want to model distributions of lung function, then that's a problem because we only have observations on a skewed part of the population and we're missing people with higher or better lung function. So in that case, the probability of data missing is conditioned on the actual outcome. So in this paper by Alexina Mason et al., they proposed three graphical models which can be incorporated in any regression question or any probabilistic graphical model to compensate for missing data. So in the first case, where data is missing at random, we could see that we have some outcome that we're trying to measure. So this could be lung function. And we have a set of features which we assume predicts this. So we want to learn the weights beta that affect this. But we also have a model of missingness. So we have P, which is the probability of data being missing or not. And we want to learn what's the distribution of P. So in the case where data is missing completely at random, the model we're interested in, which is predicting the influence of features on some parameter, which could be lung function, is independent of the model of missingness. In the case where data is missing at random, in this case, we have the distribution of P, or the probability of missingness, is affected by the things you observe. So it's affected by your feature data set. And in the case where data is not missing at random, we have a line between a conditional dependency between the outcome, so the thing we're trying to predict, and the probability of missingness. So these are very heuristic ways of thinking of missingness and incorporating them into models as well. Related to the challenge of missingness is the challenge of generalizability. So if you have a skewed population, or if you have data that's missing not at random, it's very hard to infer the distribution of the population that you haven't seen. So this is a problem in healthcare, that in healthcare we have certain populations and we're looking at distributions across those populations, and we want to take those findings and and target them to another population that we've not observed their data on. And this is a common problem in healthcare. So, for example, you can have a hospital in a particular area. You want to predict what's the probability of someone having cardiac failure in that particular hospital. You go to a hospital in another location. How can you generalize the findings from one hospital to that? So this is like having a training set and having a test set in totally different populations. But sometimes also you may not measure the same thing in that test set as well. So how do you make predictions in that test set? So this is a big challenge in healthcare, to have truly generalizable and scalable AI for healthcare. And it's a problem that's caused by missing data, inadequately sampled data, and data that doesn't represent the diversity of the population. So with generalizability, the problem we have is that training data sets are not necessarily representative of the diversity of the population or the heterogeneity of healthcare conditions. So there's a paper that which I'll present in the next few slides, that proposes using transfer learning to overcome this problem. So how can you maximize the utility of the poor data that you have, and how can you improve a model's ability to generalize as well? So this is work done from Mahela van der Shaw's lab, using transfer learning for sparsity of data. So good quality data for healthcare is expensive and often very hard to obtain. So this is just the reality of healthcare data. So the aim is to maximize information by using multiple data sources. And there are two challenges that they try to address in this paper. The first is a challenge of feature mismatch, where you may have two different data sets, and you have different features to predict a particular outcome. So the particular outcome used in this paper is cardiovascular disease, where you have one hospital, you're predicting cardiovascular disease, and you try to transfer those learnings to another hospital, but you measure different things. You take different clinical measures. How do you transfer learnings from the two, across the two? The next problem they try to address with this is distribution mismatch, where you have different populations, and in this case, they look at different demographic populations across two hospitals. So here they use a GAN architecture to efficiently enlarge the data set. So yesterday there was a talk on data augmentation. And the aim is to get better predictive models than if you simply used a particular training data set. So just at a high level, they propose this, this piece of work, which I think is something that is quite neat and quite, it's a nice concept, not just for looking at transfer learning across different populations, but also looking at missing data as well. So here you have different data domains, and these could be different hospitals. And in each of those domains, we have different feature sets, which are the X1, X2, and we have something we're trying to predict. So in the case of this paper, they're trying to predict cardiovascular disease with these different, in these different domains, but you have different feature sets. So they have an encoder F, and a latent variable Z for each of these different domains, and they have a decoder as well, which allows them to make predictions within a particular domain. So I highly recommend this paper, which was presented at ICML last year. The next challenge in healthcare, and this is the last challenge I'd look at, is that of causality, which I think is really important as well to invest time in for looking at machine learning for healthcare. So with causal reasoning, this very often motivates a lot of the questions in machine learning for healthcare. We want to know, would something work? What is the cause, or what's the nature of disease development? So before an association is assessed, we're looking for the possibility of whether something is causal or whether it's correlated. So if something is causal, then we can intervene early. So the aim in causal reasoning is to infer dynamics of belief under changing conditions. And one thing I don't have a reference to is, in these slides actually, is the whole area of counterfactual reasoning and causality. And this is a huge area of research. So I recommend here the work by David Sontag, on using counterfactual reasoning, using representation learning as a way of doing counterfactual reasoning. And the main thing we're trying to do here is say, what would have happened if we gave a particular patient a different drug? And in this paper, he goes through different scenarios for counterfactual reasoning. But here what I'm going to do is just go through some of the basic parameters for causal reasoning in healthcare data. So the first is where we're looking at biomarkers or risk factors in healthcare. So a biomarker is some biological measurement made before treatment that may indicate what is the probability of success of a particular treatment. So imagine we are in a clinical trial scenario and we have some random allocation of treatment, but there's some risk factor which affects the probability of a treatment being successful or not. So in causal reasoning, we'd want to know what are these biomarkers? Have we measured the right biomarkers? Also, there's a concept of a predictive biomarker. So there may be biomarkers that are just associative. So in this case, the biomarker is not causal, it's just associated with outcome. But we want to know, is that biomarker causal or not? So the way we model this is slightly different, and we'd say that the biomarker may be a moderator. So if you have a random allocation, your biomarker may affect the effect of treatment on a particular outcome. So in that case, we say it's a moderator, where the biomarker changes the probability of success of a particular outcome. In another case, we may say that a particular biomarker is a mediator for an outcome. And in this case, we can say that the mechanism is causal. So we want to know what's the effect of a treatment on an outcome and what proportion of that effect is mediated via some mediator. And in causal reasoning, we also want to infer some sort of latent structure as well, which may be things that we haven't directly measured, but may affect the outcomes. So an example of this of merging these together is using efficacy and mechanism evaluation to try to understand causal modeling frameworks in personalizing health. So in such a case, you may have your treatment or random allocation. You have moderators which affect the probability of treatment success, and you also have mediators and prognostic risk factors. So a lot of causality and machine learning for healthcare is looking at separating when a biomarker is a moderator, when it's possibly a mediator, or when it's possibly a risk factor, a prognostic risk factor. So an example of this taken from the literature is in cancer treatment. So in cancer treatment, for example, you may have a particular treatment, and a possible mediator may be the tumor size. So the tumor size may have an effect on survival, indirect effect on survival, but also the treatment has some direct effect on survival. And there may be genetic markers which modify the effect of treatment, and also prognostic biomarkers which don't have a direct effect on survival or may have an effect on other things. But for causal inference, it's very hard to untangle when these things are causal or not. So we can learn the structure of these graphs. But it's always good to go to the first principles, which are the Bradford-Hill principles, who were really the first people who thought, how can we say when something is causal or not? So the 6 main principles are those of plausibility. So if we see an association, is it plausible that it's causal? Consistency. So in the Bradford-Hill used the example of smoking and lung cancer, where you see that smoking consistently causes lung cancer, or asbestos consistently causes lung cancer. Temporality as well, that the cause precedes the disease. Strength of the association, so the more you smoke, the more you're likely to get lung cancer. Specificity, so does a single cause lead to a particular effect? And dose response, so the higher the exposure, the higher the risk of disease. And there's been a lot of development in the machine learning community in principles of causality. And one particular paper that I really like is this one by David Bly, looking at the blessing of multiple causes, where they're testing assumptions of causality in general, but in healthcare, as we can think of healthcare as a particular example. And one of the main challenges of inferring causality in healthcare is unmeasured confounding, so things that you haven't measured that may explain the effect of particular drugs. So in this paper, they propose a deconfounder, which is used to capture this latent structure in causality. So they make two basic assumptions, and I think the conclusion of the paper is that assuming causality is very hard. So there's one assumption that's testable, that you have a good model for the assigned causes. So if you assume some causal structure and some latent variable, which is unmeasured confounding, this is something that's testable. So you can test whether model is better or not. And the second assumption is not testable and a lot weaker, which is that there's no unobserved single cause confounders, which is a lot harder to do. So those are the main challenges for machine learning for healthcare. But I think from these challenges, there's a huge opportunity as well. And I think this is a particular opportunity, an opportunity particular to this community as well, which is the opportunity to collaborate for data collection pipelines. And I think this is the number one challenge for machine learning for healthcare, getting good data. And within Latin America, I think there's a big window of opportunity for really collecting good data sets, which are from this population as well. And this is really important. So good machine learning for healthcare starts with good study design. And the window of opportunity for curating good data sets is being involved from the beginning of curating those good machine learning or modeling data sets in order to have good studies in the future. And this is where collaboration could take place between the machine learning community and the clinical community as well. Because when we're doing machine learning for healthcare and designing studies in ML for healthcare, there are three main questions we're looking at. So what is the question or hypothesis we're trying to investigate? Is the data we have adequate to address this particular question? I think this is one of the big downfalls of looking at electronic healthcare records. So having a rich data source doesn't necessarily mean that we're going to be able to do good machine learning on that data set. And this is a really good opportunity to really think, how could we design studies that address these questions? So this is actually standard practice within a pharmaceutical setting where you are setting up studies, but this is something that's not currently done a lot in the machine learning for healthcare community, where you're starting with thinking of good design and whether the data, how can you curate data that is fit for purpose for doing these sorts of modeling strategies. So I'm just going to highlight this, having good data by design. So we need to set up studies to answer a research question, because we could design is the most important aspect of machine learning for healthcare, and perhaps the most neglected aspect of healthcare as well. So the study design should match the research question, because no matter how clever our models are, if the data isn't fit for purpose, it's very hard to do anything meaningful with that. So we don't, And in conclusion, I'd just say that context matters. So a lot of the work in machine learning for healthcare that I've presented is the result of deep collaborations across clinicians and people actually doing the modeling as well. And this is really important, to think deeply about the clinical context, to find solutions that are specific to the problem we're trying to address, and to really work with clinicians and foster discussions in an interdisciplinary setting so that we develop the bigger picture of what we're trying to model. And using a data-driven approach and domain knowledge is really important for having a problem-led approach with the patients at the center of healthcare. So I'd just like to acknowledge some of my collaborators, and thank you for your attention.
Thank you, Danielle, for this amazing talk. We have time for questions, of course. We have one there, another there.
Hi.
Hi.
So I'm going to take the opportunity to make two questions. The first one is, what is your opinion on the actual clinical effectiveness of the use of machine learning and omics data, so the recent sort of information that's being gathered regarding like molecular data? And the other one is, how do we deal with the lack of diversity on the data sets that we use in machine learning for healthcare?
All right, so I'll tackle this very good questions and very hard questions. So I think the second question, the lack of diversity of data sets, I think this is where we need to encourage different communities to actually collect data. This is something that I'm convinced about, because a lot of the open source data sets that we have are from the United States or from Europe, where you don't have diversity of population. And I think this is a big window of opportunity worldwide, and especially somewhere here in places like Africa, where you have the ability to collect data sets, but at the same time, it's a very slow process. We can think that example in the first slide in the US, where in 2008, they had very little electronic healthcare records, and they were able to scale that within a period of 7 years. I think something similar can happen in different communities as well. It's a lot easier to grow these different data sets. But I think the most effective way of really developing diverse data sets is starting small and really finding what are the important problems in healthcare to solve, and curating these data sets as well. And this is a very slow process. It could be a process of many years. But this is something that I've actually seen, that it is a worthwhile investment. So the study I presented in Manchester, so I was working in that in 2008, was the first time somebody worked on that, which was myself. But that was a study that started in 1991. So you think after 17 years, you have this really well-curated data set. And it wasn't just Manchester. There were 5 other parts of the UK that did this. I think this is a really worthwhile investment, because healthcare problems don't go away, but we need to understand things very deeply. So the second question of omics data, and this is a very hard problem. So there is some really good work by Kim Anla Kao. So she's a professor out in Melbourne, which she looks a lot at these problems of how to find structure in combining different omics data sets. My opinion is that omics data is very noisy. I think part of it is probably I don't know why it's so noisy, actually. But yeah, it's problematic to find structure. But Kim Anla Kao has found some success there, so I could give you the references after that.
I was wondering if maybe there is one issue that connects all your 3 challenges that you mentioned here, is a compliance of individual to the treatment that you assign. So in your asthma study, for example, if someone who is having adverse effects from a treatment, he drops the treatment or doesn't comply with it, maybe you will not see the heterogeneity in the population that you would like to see in your data. And then it connects also with the causal analysis that you need to see. And if I may, one second question is, how did you find your 5 subtypes? Did you use Dirichlet process in the asthma?
Yes,
study?
so it was a similar process to the work on the allergic march as well, where we had some prior, and we assumed between 2 to 15 different subtypes, and just assigned children to the subtype that had the highest probability. And once we got those numbers of subtypes, then looking at this, at these trajectories over time. So I think that's a very hard question. This is one of the challenges of healthcare as well, that I haven't had there, which is the whole challenge of unmeasured confounding. I would say it's a challenge of unmeasured confounding, where you don't have compliance to treatment. And I guess in machine learning for healthcare, this is a simplifying assumption that you make, that you assume that the patient is compliant to the treatment. There are studies that measure this now. So, for example, with asthma and with COPD, for clinical trials, now what they do is they have in the device, they can tell whether a patient actually uses a drug or not. But even then, sometimes people don't like to use a drug, so they may just spray it and not spray it within their mouth or whatever. So this is a huge challenge that, yeah, how do we address it? That's a very, very interesting problem. But I do agree that is an underlying challenge of everything I presented, and that's why latent variable modeling, what we're trying to do is reduce the uncertainty in these measurements as well. Yeah, it's a huge thing.
More questions for the public here on the floor?
Hello. So thank you for your talk. I have one specific question, which is, so to put a little context, we here have a kind of, we're starting with a multidisciplinary approach to this kind of problem, together with doctors and physicians. And we had the visit of a Brazilian epidemiologist named Marcelo Barreto, who has set up an amazing hardware and software infrastructure for gathering and curating data. And it's been a huge investment he's done. We were quite impressed by his work. I was wondering whether Microsoft, with its huge capabilities, is doing something similar. So there's something I forgot, which is the main problem that this kind of the kind of data gathering process that these people we have here is the regulations on privacy, and it's taken very seriously by many governments. So there is some protocols for using the data without accessing the data in some way. And I was wondering whether you have the same problems in England, or whether these are being addressed by some specific protocols or infrastructures.
Very hard question, but very important question. Yes, so I think, all right, so machine learning for healthcare, a lot of the tech companies are looking at ways of having privacy preserving modeling, and I think this is a standard. How can you deploy? I think it's especially important when you're thinking of deploying models, but especially in machine learning for health. Well, a lot of areas that use data, you're using private data. So how do you run algorithms in a privacy-preserving way? So there are things that are in place for doing this that are standard. Yeah, and I think it's super important that you do have these regulations that are in place, and it's good that the tech community is catching up with that. So they are thinking of ways, how do you run these algorithms in a privacy-preserving way? But I would say it's hard. It's a hard problem as well. And because it's a hard problem, it does limit what you are able to do as well in terms of data, I think. But yeah, I'd like to talk more about this offline, but yeah, I think it's a good problem.
Sure.
Yeah.
Thank you very much.
Hi, Daniel. Thank you for your talk. Very interesting. I'm quite curious about how do you deal with the scarcity of samples, because you deal with humans in general, and you need an ethics committee, and it's not as easy as pictures of dog and cats, for example. And at the same time, with the high-dimensional environment, I mean, if you work with multi-omic data or with other type of complex data from biology or biomedicine, And how can you deal with that constraint and trying to generalize as well as the other examples that we have been listening during these days?
Yeah, so the first thing that I tend to do is identifying what are the set of problems you're trying to look at. So these could be hypotheses if you want to do something that's more hypothesis testing. The second thing is power calculations. So just looking, do I have sufficient power in my study or sufficient sample size in my study to find the effects that I'm looking for? So doing power calculations is actually quite challenging. I think it's probably one of the most challenging parts of working with healthcare data, because it means eliciting values from a clinician. So it's a big communication process in trying to get a clinician or the person who is generating the data to give an idea of how variable they think the data is. So getting them to give estimates of standard deviation variance. But I think this is important if we have small data sets, because sometimes it's very limited what inference you can make, especially if you want to make assumptions of causality. If the data is too sparse, maybe you just can't do it. And it's a bit of a painful process sometimes to just let something go and say, you can't do it. But sometimes it may be a matter of being able to collect more data, or if you have sparse data, you can use it as a proof of concept. So you can use it as a pilot to develop something further. But I think that stage of doing a power calculation just to see what is the sample I need in order to make statistically meaningful conclusions is a really important step. And this also informs good study design as well. But yeah, that's a good question, actually. I must say these questions are very challenging, but very, very, very good questions to think about as well. Really like that.
Yeah, that's it. Thanks again, Danielle, for this
Thank
amazing
you.
talk.